Important questions of BZYCT 135. Q. Difference between the following. 1. Difference between blood and lymph. Blood is red due to the presence of hemoglobin. Lymph is pale yellow and lacks hemoglobin. Blood transports oxygen, nutrients, and hormones. Lymph transports tissue fluid, waste, and immune cells. Asterisk. Blood contains red blood cells, RBCs, white blood cells, WBCs, and platelets. Lymph contains only white blood cells. Asterisk. Blood flows through arteries and veins. Lymph flows through lymphatic vessels. Asterisk. Blood clots quickly due to platelets. Asterisk. Lymph clots slowly and less efficiently. 2. Difference between artery and vein. Arteries carry blood away from the heart. Veins carry blood toward the heart. Arteries usually carry oxygen-rich blood, except pulmonary artery. Veins usually carry oxygen-poor blood, except pulmonary vein. Arteries have thick, elastic muscular walls. Veins have thin and less elastic walls. Arteries do not contain valves. Veins contain valves to prevent backflow. Blood pressure in arteries is high. Blood pressure in veins is low. 3. Difference between ureotelic and uricotelic organisms. Ureotelic organisms excrete urea. Uricotelic organisms excrete uric acid. Urea is soluble in water. Uric acid is almost insoluble. Ureotelism results in moderate water loss. Uricotelism results in minimal water loss. Ureotelic organisms include humans and amphibians. Uricotelic organisms include birds and reptiles. Ureotelism requires moderate energy. Asterisk. Uricotelism requires high energy. 4. Difference between asexual and sexual reproduction. Asexual reproduction does not involve gametes. Sexual reproduction involves male and female gametes. Asexual reproduction produces genetically identical offspring. Sexual reproduction results in genetic variation. Asexual reproduction occurs through binary fission, budding, etc. Sexual reproduction involves fertilization. Asexual reproduction is a fast process. Sexual reproduction is relatively slow. Examples of asexual reproduction: Hydra, amoeba. Examples of sexual reproduction: humans, animals. 5. Difference between hydrolases and lyases. Hydrolases break bonds using water. Lyases break bonds without using water. Hydrolases catalyze hydrolysis reactions. Lyases catalyze elimination or addition reactions. Examples of hydrolases: amylase, protease. Examples of lyases: fumarase, aldolase. Hydrolases act on esters, peptides, and glycosides. Lyases act on carbon-carbon and carbon-nitrogen bonds. Hydrolases require water molecules. Lyases do not need water. 6. Difference between A band and I band of myofibrils. A band appears dark under a microscope. I band appears light. A band contains both actin and myosin filaments. I band contains only actin filaments. A band remains unchanged during muscle contraction. I band shortens during contraction. A band is located in the center of the sarcomere. I band lies on both sides of the A band. A band helps in generating force. I band contributes to muscle elasticity. 7. Difference between open and closed circulatory system. In open circulation, blood is not entirely in vessels. In closed circulation, blood is confined within blood vessels. Open systems are less efficient in oxygen and nutrient transport. Closed systems are more efficient. Blood pressure is low in open systems. Blood pressure is high in closed systems. Open systems have hemolymph instead of blood. Closed systems have blood and lymph. Open systems are found in insects and mollusks. Closed systems are found in humans, fish, and vertebrates. 8. Difference between water-soluble and fat-soluble vitamins. Water-soluble vitamins are not stored in the body. Fat-soluble vitamins are stored in the liver and fatty tissues. Water-soluble vitamins include B complex and vitamin C. Fat-soluble vitamins include A, D, E, and K. Water-soluble vitamins are absorbed with water. Fat-soluble vitamins require dietary fats for absorption. Overdose of water-soluble vitamins is rare. Overdose of fat-soluble vitamins can be toxic. Deficiency symptoms appear quickly in water-soluble vitamins. Deficiency symptoms appear slowly in fat-soluble vitamins. 9. Difference between glycogenin and glycogen synthase. Glycogenin initiates glycogen synthesis. Glycogen synthase elongates the glycogen chain. Glycogenin is a protein. Glycogen synthase is an enzyme. Glycogenin forms a primer for glycogen. Glycogen synthase requires a primer to function. Both function in the cytoplasm. Glycogenin has self-glucosylating activity. Glycogen synthase adds glucose molecules to the chain. Glycogenin is the core protein of glycogen particles. 10. Difference between epinephrine and norepinephrine. Epinephrine is secreted by the adrenal medulla. Norepinephrine is secreted by both adrenal medulla and neurons. Epinephrine functions as a hormone. Norepinephrine functions mainly as a neurotransmitter. Epinephrine increases heart rate and dilates airways. Norepinephrine increases heart rate and constricts blood vessels. Epinephrine prepares the body for fight or flight response. Norepinephrine maintains blood pressure. Epinephrine is used in asthma treatment. Norepinephrine is used to treat low blood pressure. 11. Difference between aminotelic and ureotelic organisms. Aminotelic organisms excrete ammonia. Ureotelic organisms excrete urea. Ammonia is highly toxic and requires dilution. Urea is less toxic and more stable. Aminotelic excretion requires a large amount of water. Ureotelic excretion needs moderate water. Aminotelism is common in aquatic animals like fish. Ureotelism occurs in mammals and amphibians. Aminotelism uses less energy. Ureotelism uses more energy than aminotelism. Q. Explain term. Pheromone. Pheromones are chemical substances secreted by animals or humans that influence the behavior or physiology of others of the same species. They are often used for communication, including attracting mates, marking territory, or signaling danger. Insects like ants use trail pheromones to guide others to food sources. Pheromones can be airborne or deposited on surfaces. Human pheromones are still under scientific investigation, with some evidence suggesting subtle behavioral effects. They may affect reproductive behavior, mother-infant bonding, and social interactions. Some animals release alarm pheromones to alert others of predators. Pheromones act at very low concentrations and can travel long distances. They are detected by specialized sensory cells, especially in the olfactory system. Synthetic pheromones are used in pest control by disrupting mating patterns. Residual volume. Residual volume is the amount of air remaining in the lungs after a maximal exhalation. It prevents the lungs from collapsing by maintaining alveolar inflation. This volume ensures continuous gas exchange between breaths. In a healthy adult, it is approximately 1,200 milliliters. It cannot be measured directly using spirometry. Residual volume contributes to the total lung capacity. It is essential for maintaining partial pressure of gases in the lungs. This volume increases with age and certain lung diseases like emphysema. It helps prevent atelectasis, a condition where the lungs collapse. It is an important parameter in pulmonary function testing. Residual volume is the amount of air remaining in the lungs after a maximal exhalation. Osmoregulation. Osmoregulation is the process of maintaining water and salt balance in an organism. It ensures proper osmotic pressure in body fluids. The kidneys are the primary organs responsible for osmoregulation in humans. Antidiuretic hormone, ADH, regulates water reabsorption in the kidneys. Marine fish drink seawater and excrete salt through gills to maintain balance. Freshwater fish excrete dilute urine and absorb salts through their gills. Plants use vacuoles and osmotic adjustments to maintain water balance. Dehydration and overhydration are both consequences of osmoregulatory failure. Insects use Malpighian tubules for osmoregulation. Homeostasis heavily relies on efficient osmoregulation. Oogenesis. Oogenesis is the process of forming female gametes, ova, in the ovaries. It begins before birth and completes. Only after fertilization. Oogonia multiply by mitosis and develop into primary oocytes. Primary oocytes begin meiosis but are arrested in prophase I until puberty. At puberty, one oocyte resumes meiosis during each menstrual cycle. The secondary oocyte is released during ovulation. Meiosis II completes only if fertilization occurs. Each oogenesis cycle results in one ovum and polar bodies. It contrasts with spermatogenesis, which produces 4 sperms per cycle. Oogenesis is regulated by hormones like FSH and LH. Parthenogenesis. Parthenogenesis is a form of asexual reproduction where offspring are produced from unfertilized eggs. It can be observed in insects, amphibians, reptiles, and some birds. Parthenogenesis may be obligate, always occurs, or facultative under specific conditions. It leads to the formation of genetically identical offspring. Honeybees exhibit parthenogenesis where drones develop from unfertilized eggs. In some species, environmental stress can trigger parthenogenesis. It is a mechanism for rapid population increase. Some reptiles like Komodo dragons reproduce via parthenogenesis. It bypasses the need for male gametes. Parthenogenesis has applications in cloning and reproductive research. Parthenogenesis is a form of asexual reproduction where offspring are produced from unfertilized eggs. It can be observed in insects, amphibians, reptiles, and some birds. Parthenogenesis may be obligate, always occur. Monosaccharide. Monosaccharides are the simplest carbohydrates with a single sugar unit. They are the building blocks of disaccharides and polysaccharides. Examples include glucose, fructose, and galactose. They are easily soluble in water and sweet to taste. Monosaccharides are categorized by the number of carbon atoms triose, pentose, hexose. They play a central role in cellular respiration. Glucose is the primary energy source in living organisms. They can exist in linear or ring structures. They are reducing sugars due to free aldehyde or ketone groups. They serve as precursors for biosynthetic pathways. Monosaccharides are the simplest carbohydrates with a single sugar unit. They are the building blocks of disaccharides and polysaccharides. Examples include glucose, fructose, and galactose. They are easily soluble in water and sweet to taste. Monosaccharides are categorized by the number of carbon atoms triose, pentose, hexose. They play a central role in cellular respiration. Glucose is the primary energy source in living organisms. They can exist in linear or ring structures. They are reducing sugars due to free aldehyde or ketone groups. They serve as precursors for biosynthetic pathways. Monosaccharides are the simplest carbohydrates with a single sugar unit. They are the building blocks of disaccharides and polysaccharides. Examples include glucose, fructose, and galactose. Allosteric enzymes. Allosteric enzymes have an active site and one or more regulatory sites. Their activity is modified by molecules binding to allosteric sites. These regulators may act as activators or inhibitors. Allosteric regulation helps in fine-tuning enzyme activity. They often show sigmoidal, S-shaped kinetics instead of hyperbolic. ATP can act as an allosteric effector. Enzymes involved in metabolic control like phosphofructokinase are allosteric. They play a role in feedback inhibition. Cooperative binding is a key feature of allosteric enzymes. They are essential for regulating metabolic pathways efficiently. They play a role in feedback inhibition. Neurotransmitter. Neurotransmitters are chemical messengers used by neurons to communicate. They are released from synaptic vesicles into the synaptic cleft. They bind to specific receptors on postsynaptic cells to elicit responses. Examples include dopamine, serotonin, acetylcholine, and norepinephrine. Their action is terminated by reuptake, enzymatic degradation, or diffusion. They play roles in mood, cognition, motor control, and autonomic function. Neurotransmitter imbalances are associated with diseases like depression, Parkinson's, and schizophrenia. They are classified as excitatory or inhibitory. Some drugs mimic or block neurotransmitters, agonists and antagonists. Synaptic plasticity depends on neurotransmitter dynamics. Tidal volume. Tidal volume is the amount of air inhaled or exhaled during a normal breath. In adults, it averages about 500 milliliters. It varies based on age, body size, and physical activity. Tidal volume is measured during pulmonary function tests. It increases during deep breathing or exercise. It is a component of vital capacity. It ensures basic oxygen supply to the body at rest. Reduced tidal volume may occur in restrictive lung diseases. Tidal volume combined with respiratory rate gives minute ventilation. It is vital for maintaining proper blood gas levels. Tidal volume is the amount of air inhaled or exhaled during a normal breath. In adults, it averages about 500 milliliters. It varies based on age, body size, and physical activity. Isoenzyme. Isoenzymes or isozymes are different molecular forms of the same enzyme. They catalyze the same reaction but differ in structure or kinetic properties. They may be expressed in different tissues or developmental stages. Lactate dehydrogenase (LDH) has 5 isoenzymes used in diagnostics. Isoenzymes help in tissue-specific regulation of metabolism. Creatine kinase isoenzymes are markers for myocardial infarction. They allow fine-tuned enzyme activity in varied conditions. Their study is useful in evolutionary and comparative biology. They often differ in pH optima and substrate affinity. Isoenzymes provide biochemical diversity within a species. Respiration. Respiration is the biochemical process of breaking down glucose to release energy. It can be aerobic with oxygen or anaerobic without oxygen. In aerobic respiration, glucose is fully oxidized to carbon dioxide and water. It involves glycolysis, Krebs cycle, and electron transport chain. ATP is the energy currency generated during respiration. Anaerobic respiration produces less ATP and byproducts like lactic acid or ethanol. Mitochondria are the powerhouse of aerobic respiration. Respiration supports all energy-dependent functions like muscle contraction, active transport, and biosynthesis. Oxygen acts as the final electron acceptor in the electron transport chain. Respiratory rate changes with physical activity, stress, and metabolic demand. Q. Short notes on 1. Allosteric enzymes. Definition. Allosteric enzymes are enzymes that change their shape and activity when a molecule binds to a site. Other than the active site, this site is known as the allosteric site. Mechanism. The binding of an effector, activator, or inhibitor at the allosteric site induces a conformational change. This change can either increase positive regulation or decrease negative regulation, the enzyme's activity. Types of effectors. Allosteric activators enhance enzyme activity. Allosteric inhibitors reduce or inhibit enzyme activity. Example: aspartate transcarbamylase, ATCase in the synthesis of pyrimidine nucleotides. Significance. Regulates metabolic pathways. Helps maintain homeostasis in the cell. Responds quickly to changes in the environment. 2. Neurotransmitter. Definition. Neurotransmitters are chemical messengers that transmit signals across synapses between neurons, or from neurons to muscles, glands. Types. Excitatory neurotransmitters increase the chance of the neuron firing, e.g., glutamate. Inhibitory neurotransmitters decrease the chance of firing, e.g., GABA. Major neurotransmitters. Dopamine, reward, pleasure, movement. Serotonin, mood, appetite, sleep. Acetylcholine, muscle activation, learning. Norepinephrine, alertness, arousal. Synaptic transmission. Neurotransmitter is released from presynaptic neuron. Binds to receptors on the postsynaptic cell. Triggers a response or action potential. Importance. Fundamental in brain function and behavior. Target of many drugs and medications, e.g., antidepressants, anesthetics. 3. Residual volume. Definition. Residual volume, RV, is the amount of air remaining in the lungs after a maximal exhalation. Typical value. Around 1200 ml in adult males and 1000 ml in females. Function. Prevents lung collapse by keeping alveoli open. Maintains continuous gas exchange even between breaths. Measurement. Cannot be measured directly by spirometry. Measured using body plethysmography or gas dilution methods. Clinical relevance. Changes in RV may indicate lung diseases, e.g., emphysema, asthma. 4. Tidal volume. Definition. Tidal volume (TV) is the volume of air moved into or out of the lungs during a normal, resting breath. Typical value. Around 500 milliliters in healthy adults. Components. Includes both alveolar air used in gas exchange and dead space air not used. Function. Supplies oxygen and removes carbon dioxide from the bloodstream. Changes with activity. Increases with exercise or stress. Decreases during shallow breathing. 5. Isoenzyme, isozyme. Definition. Isoenzymes are different molecular forms of the same enzyme that catalyze the same reaction but vary in structure and location. Characteristics. Encoded by different genes. Different kinetic properties. Different tissue distributions. Examples. Lactate dehydrogenase LDH found in heart, liver, and muscles. Creatine kinase CK used in diagnosing heart attacks. Significance. Useful in clinical diagnostics. Helps adapt enzyme function to specific tissue needs. 6. Respiration. Definition. Respiration is the process by which cells break down glucose to release energy, ATP. Types. Aerobic respiration with oxygen. Glucose plus O right pointing arrow CO plus HO plus ATP. Anaerobic respiration without oxygen. Glucose right pointing arrow lactic acid plus ATP in animals. Or glucose right pointing arrow ethanol plus CO plus ATP in yeast. Stages in aerobic respiration. Glycolysis in cytoplasm. Krebs cycle, citric acid cycle in mitochondria. Electron transport chain, mitochondrial inner membrane. Importance. Provides energy for cellular processes. Maintains life functions. 7. Blood. Definition. Blood is a fluid connective tissue that circulates throughout the body delivering oxygen and nutrients, and removing waste. Components. Plasma 55% water, proteins, hormones, nutrients. Formed elements 45%. Red blood cells RBCs carry oxygen. White blood cells WBCs immune response. Platelets, clotting. Functions. Transport gases, nutrients, hormones. Immune defense. Temperature regulation. Waste removal. Blood types. A, B, AB, O, based on antigen presence. Rh factor plus or minus is also important in compatibility. 8. Water-soluble vitamins. Definition. These vitamins dissolve in water and are not stored in large quantities in the body. List of water-soluble vitamins. Vitamin C, ascorbic acid. B complex group. B1, thiamine, B2, riboflavin, B3, niacin, B5, pantothenic acid, B6, pyridoxine, B7, biotin, B9, folate, B12, cobalamin. Functions. Coenzymes in metabolism. DNA synthesis. Nervous system support. Deficiency diseases. B1 beriberi. B3 pellagra. B12 pernicious anemia. C. Scurvy. Excretion. Excess is excreted in urine. 9. Macronutrient. Definition. Macronutrients are nutrients required in large quantities for energy and bodily functions. Types. Carbohydrates, main energy source. Proteins, for growth, repair. Enzymes. Fats, energy storage, hormone production. Water, transport, temperature regulation. Daily requirements. Vary by age, gender, activity level. Energy values. Carbohydrates, 4 kilocalories per gram. Proteins, 4 kilocalories per gram. Fats, 9 kilocalories per gram. Importance. Balanced intake supports health, growth, and metabolism. 10. Essential vitamins. Definition. Vitamins that are crucial for various metabolic functions and must be obtained from the diet. Categories. Fat-soluble, A, D, E, K. water-soluble, B-complex and C. Key roles: vitamin A, vision, immunity. Vitamin D, calcium absorption. Vitamin E, antioxidant. Vitamin K, blood clotting. Deficiency issues: A. Night blindness. D. Rickets, osteomalacia. E. Neurological issues. K. Excessive bleeding. 11. Emulsification. Definition: emulsification is the process of breaking down large fat globules into smaller droplets. Where it happens: in the small intestine during digestion. Role of bile: bile salts from the liver emulsify fats, increasing surface area for lipase enzymes. Why it's important: enhances fat digestion, allows absorption of fatty acids and fat-soluble vitamins. Analogy: like mixing oil and water using soap, bile acts like a detergent. Q. Answer the following questions. 1. Glycogenesis and glycogenolysis, approximately 120 lines. Large diamond glycogenesis. Glycogenesis is the process of converting glucose into glycogen for storage in the liver and muscles. It occurs when blood glucose levels are high, such as after a carbohydrate-rich meal. The process is anabolic, building up, and is regulated by insulin. Steps in glycogenesis. Glucose phosphorylation. Glucose is phosphorylated to glucose 6-phosphate, G6P, by hexokinase or glucokinase. Isomerization. G6P is converted to glucose-1-phosphate by phosphoglucomatase. Activation. Glucose-1-phosphate reacts with UTP to form UDP glucose, catalyzed by UDP glucose pyrophosphorylase. Glycogen synthesis. Glycogen synthase adds UDP glucose to a pre-existing glycogen primer. Branching. Branching enzyme forms alpha-1, 6 linkages to increase solubility and storage capacity. Hormonal regulation. Insulin stimulates glycogenesis. Glucagon and epinephrine inhibit it. Significance. Prevents hyperglycemia. Provides energy reserves during fasting or exertion. Large diamond glycogenolysis. Glycogenolysis is the catabolic process of breaking down glycogen into glucose-1-phosphate and free glucose to maintain blood glucose levels during fasting or exercise. Steps in glycogenolysis. Phosphorylysis. Glycogen phosphorylase removes glucose residues as glucose-1-phosphate. Debranching. Debranching enzyme transfers short chains and removes the last glucose from branches. Conversion. Glucose-1-phosphate is converted to glucose 6-phosphate by phosphoglucomatase. Hormonal regulation. Glucagon and epinephrine stimulate glycogenolysis. Insulin inhibits it. Significance. Maintains blood glucose level. Provides energy during fasting or exercise. 2. Extrinsic and intrinsic pathways, blood clotting. Large diamond intrinsic pathway. Triggered by trauma inside the vascular system. Slower but more complex. Initiated by exposure of blood to negatively charged surfaces, like collagen. Involves clotting factors 12, 11, X, 8. Activated partial thromboplastin time, APTT, measures this pathway. Large diamond extrinsic pathway. Triggered by external trauma that causes blood to escape from the vascular system. Faster response. Initiated by tissue factor 3 or thromboplastin, released from damaged tissues. Involves factor 7 and tissue factor. Measured by prothrombin time, PT. Large diamond common pathway. Both pathways converge at factor X. Factor X converts prothrombin 2 into thrombin. Thrombin converts fibrinogen into fibrin to form the clot. Significance. Maintains hemostasis. Prevents excessive bleeding. Balances clot formation and dissolution. 3. Allosteric enzymes and isoenzymes. Large diamond allosteric enzymes. Enzymes that are regulated by molecules binding at sites other than the active site, allosteric sites. Display sigmoidal kinetics instead of hyperbolic. Often involved in metabolic regulation. Examples. Aspartate transcarbamylase. PHOSPHOFRUCTOKINASE1, PFK1. Types of effectors. Positive, activators. Negative, inhibitors. Functions. regulate metabolic pathways, allow feedback inhibition, act as biological switches. Large diamond isoenzymes, isozymes. Different molecular forms of an enzyme that catalyze the same reaction. Differ in amino acid sequence, kinetic properties, and tissue distribution. Examples. Lactate dehydrogenase LDH has 5 isoforms LDH1 to LDH5. Creatine kinase CK, CKM, CKMB, CKBB. Clinical importance. LDH1 is elevated in myocardial infarction. CKMB used as a marker for heart damage. 4. Osmoconformers and osmoregulators. Large diamond osmoconformers. Organisms that maintain internal osmolarity equal to their environment. Mostly marine invertebrates and some fish. No energy spent on osmoregulation. Examples. Jellyfish, marine crabs. Large diamond osmoregulators. Maintain a constant internal osmotic pressure despite external changes. Found in freshwater and terrestrial animals. Examples. Humans, freshwater fish, birds. Key differences. Feature osmoconformers, osmoregulators. Energy use low-high. Internal environment matches surroundings, remains constant. 5. Cilia and flagella. Large diamond cilia. Short, hair-like structures. Numerous per cell. Move rhythmically to sweep fluid or particles. Found in respiratory tract, fallopian tubes. Large diamond flagella. Long, whip-like structures. Few in number, often 1 to 2. Move in an undulating or propeller motion. Found in sperm cells. Structure, common. 9+2 arrangement of microtubules. Powered by ATP and dynein arms. 6. Skeletal and cardiac muscle. Feature skeletal muscle, cardiac muscle. Location attached to bones, heart walls. Control voluntary, involuntary. Appearance striated. Nuclei multinucleated, single nucleus. Intercalated discs absent, present. Contraction type rapid, short-lived, rhythmic and continuous. Functions. Skeletal, body movement, posture. Cardiac, pump blood throughout the body. 7. Composition of blood. Blood is a connective tissue composed of large diamond plasma 55%, water 90%, proteins albumin, globulin, fibrinogen, nutrients, hormones, waste. Large diamond formed elements 45%. Red blood cells RBCs carry oxygen using hemoglobin. White blood cells WBCs fight infection, lymphocytes, neutrophils, monocytes, eosinophils, basophils. Platelets help in blood clotting. Functions. Transport gases and nutrients. Immune defense. Clotting and wound healing. Regulation of body temperature and pH. 8. Significance of Km and Vmax in enzyme reactions. Large diamond Km, Michaelis constant. Substrate concentration at which reaction rate is half of Vmax. Indicates enzyme's affinity for substrate. Low Km equals high affinity. High Km equals low affinity. Large diamond Vmax. Maximum rate of enzyme catalyzed reaction. Occurs when all active sites are saturated. Michaelis-Menten equation. V equals Vmax S Km plus S V equals Km plus S Vmax. S. Importance. Enzyme kinetics in pharmacology. Disease diagnosis. Biotechnological applications. Q1. Draw a labeled diagram of cardiac cycle and explain all the events associated with one heartbeat. Answer. Cardiac cycle refers to the sequence of events in one heartbeat, lasting about 0.8 seconds. Phases. Atrial systole, 0.1 seconds, atria contract, right pointing arrow, blood flows into ventricles. Ventricular systole, 0.3 seconds, ventricles contract, right pointing arrow, AV valves close, first heart sound, lub, right pointing arrow, blood is pumped into aorta and pulmonary artery. Complete diastole, 0.4 seconds, all chambers relaxed, right pointing arrow, blood flows from veins into atria, right pointing arrow, semilunar valves close, second heart sound, dub. Diagram. Includes 4 chambers RA, RV, LA, LV. Valves: tricuspid, bicuspid, mitral, pulmonary and aortic valves. Arrows showing blood flow. Additional points. SA node, pacemaker, initiates electrical impulse. AV node, bundle of His, Purkinje fibers, spread the impulse. ECG reflects electrical events. One cardiac cycle pumps about 70 milliliters blood, stroke volume. Q2, discuss the significance of Km and Vmax. Answer. Km, Michaelis constant, substrate concentration at which the enzyme works at half of its maximum speed. Vmax, maximum rate of an enzyme-catalyzed reaction when substrate is abundant. Significance. Low Km right pointing arrow, high enzyme substrate affinity. High Km right pointing arrow, low affinity. Helps determine enzyme efficiency and reaction speed. Useful in drug design and metabolic studies. Lineweaver-Burke plot is used to calculate Km and Vmax graphically. Q3. Explain the components of electron transport chain with the help of a suitable diagram. Answer. The electron transport chain, etc., is in the inner mitochondrial membrane, key for ATP production via oxidative phosphorylation. Components. Complex I, NADH dehydrogenase, transfers electrons from NADH to coke. Complex II, succinate dehydrogenase, transfers electrons from FADH to coke. Coenzyme Q, ubiquinone, mobile carrier between complex I, II and III. Complex III, cytochrome BC, passes electrons to CYT circa. Cytochrome C, mobile electron carrier. Complex IV, cytochrome C oxidase, transfers electrons to O. Right pointing arrow forms HO. ATP synthase, complex V, uses proton gradient to generate ATP. Diagram. Show mitochondrion with all complexes, arrows for E and H flow, and ATP synthesis. Q4. What is electron transport chain, etc.? Explain etc. I with the help of schematic diagram only. Answer. Etc. is a series of protein complexes that transfer electrons from NADH, FAD to O, generating a proton gradient used by ATP synthase. Etc. I, complex I, NADH dehydrogenase. Accepts electrons from NADH. Electrons flow through FMN and Fe-S centers. Transfers electrons to coenzyme Q, ubiquinone. Pumps 4 H ions into intermembrane space per NADH. Right pointing arrow creates proton gradient. Schematic diagram should show NADH right pointing arrow, FMN right pointing arrow, Fe-S right pointing arrow, coke. Proton H pumping across membrane. Electron flow toward coke. Write short notes on checkmark Q1. How does short-term regulation of urea cycle differ from long-term regulation? Opening parenthesis. Writing hand introduction to urea cycle. The urea cycle, also known as the ornithine cycle, is a metabolic pathway that converts toxic ammonia into urea for excretion in the urine. It occurs primarily in the liver. Urea formation is crucial for detoxifying ammonia produced from amino acid metabolism. Anti-clockwise arrows. Types of regulation. Urea cycle is regulated at two levels. Short-term regulation. Long-term regulation. KeyCap 1. Short-term regulation of urea cycle. Test tube definition. It refers to the rapid changes in urea cycle activity that occur in response to immediate metabolic needs, within minutes to hours. Gear mechanism. Regulated primarily by allosteric activation of carbamoyl phosphate synthetase I, CPSI, the rate-limiting enzyme of the cycle. Key regulator. N-acetylglutamate, NAG. Microscope role of NAG. NAG is an essential allosteric activator of CPSI. It is synthesized from glutamate plus acetyl-CoA. Right pointing arrow NAG. NAG levels increase when amino acid levels are high, e.g., after protein-rich meal. Brain example. After a high-protein meal. Up pointing arrow amino acids. Right pointing arrow up pointing arrow glutamate. Right pointing arrow up pointing arrow NAG. Right pointing arrow up pointing arrow CPSI activity. Right pointing arrow up pointing arrow urea production. Stopwatch timing. Occurs rapidly, within minutes to few hours. Pushpin summary of short-term regulation. Feature short-term regulation. Main enzyme CPSI. Activator N-acetylglutamate, NAG. Stimulus high protein intake. Time minutes to hours. Purpose immediate detox of excess ammonia. KeyCap2 long-term regulation of urea cycle. Test tube definition. Involves changes in gene expression and enzyme levels over a longer period, hours to days. DNA mechanism. Increases or decreases the synthesis of urea cycle enzymes depending on diet and physiological state. Brain triggers. High protein diet. Starvation. Chronic acidosis. Prolonged fasting. Test tube enzymes involved. Carbamoyl phosphate synthetase I, CPSI. Ornithine transcarbamylase, OTC. Argininosuccinate synthetase. Argininosuccinate lyase. Arginase. Upward trend example. Prolonged high protein diet or starvation. Uppointing arrow demand for amino acid breakdown. Uppointing arrow mRNA levels for urea cycle. Enzymes. Uppointing arrow enzyme synthesis. Uppointing arrow urea cycle activity. Stopwatch timing. Occurs over hours to days or chronically. Pushpin summary of long-term regulation. Feature long-term regulation. Mechanism gene expression. Stimulus prolonged protein intake or starvation. Time hours to days. Purpose adaptive metabolic regulation. Magnifying glass differences between short-term and long-term regulation. Feature short-term regulation long-term regulation. Speed minutes to hours hours to days. Mechanism allosteric activation via NAG gene expression changes. Enzyme focus CPSI all urea cycle enzymes. Trigger protein-rich meal chronic protein intake starvation. Reversibility quickly reversible slowly reversible. Function immediate response. Adaptive response. Brain memory aid. Short-term equals switch NAG. Long-term equals factory upgrade enzyme synthesis. Book conclusion. The urea cycle's regulation is a perfect example of how our body balances metabolic flexibility and stability. Short-term regulation provides quick response, while long-term regulation offers sustained adaptation. Both are crucial for nitrogen homeostasis and prevention of hyperammonemia. Ensures enzyme levels are sufficient for long-term nitrogen disposal. Comparison table. Feature short-term regulation long-term regulation. Time scale minutes to hours hours to days. Mechanism allosteric activation e.g., NAG gene transcription and translation. Enzyme activity modified temporarily changed through new synthesis. Key regulator N-acetylglutamate glucagon, cortisol. Response to immediate nitrogen load sustained protein intake, starvation. Q2. What are the factors affecting the rate of enzyme action? Enzyme action basics. Enzymes are biological catalysts that speed up chemical reactions by lowering activation energy. Their activity can be influenced by various internal and external factors. Major factors. 1. Temperature. Optimal range. Enzymes work best within specific temperatures, usually 37 degrees Celsius for humans. Too low. Molecules move slowly right pointing arrow low collision rate. Too high. Enzyme denatures, loses shape. Upward trend graph. Bell-shaped curve. Max activity at optimum temperature. 2. pH level. Each enzyme has an optimum pH e.g., pepsin, pH 2, trypsin, pH 8. Changes in pH can disrupt ionic bonds, alter active site shape. Upward trend graph. Peak at optimal pH. Activity drops at both acidic and basic extremes. 3. Substrate concentration. Up pointing arrow substrate right pointing arrow up pointing arrow reaction rate until enzyme is saturated. At saturation, all active sites occupied equals maximum rate Vmax. Upward trend graph. Hyperbolic curve Michaelis-Menten kinetics. 4. Enzyme concentration. More enzymes right pointing arrow more active sites right pointing arrow higher reaction rate. Limited by substrate availability. 5. Inhibitors. Competitive inhibitors. Bind active site right pointing arrow reduce rate. Non-competitive inhibitors. Bind elsewhere right pointing arrow alter shape. Irreversible inhibitors. Permanently deactivate enzymes. 6. Cofactors and coenzymes. Some enzymes require Cofactors metal ions like Zn squared, Mg squared. Coenzymes organic molecules like NAD, FAD. Absence equals reduced or no activity. 7. Product concentration. Product accumulation can cause feedback inhibition. Prevents overproduction. 8. Presence of activators. Some enzymes need activator molecules to function efficiently. Q3. How amino acid reacts as acid and base. Introduction. Amino acids are amphoteric. They act as both acids and bases. They contain amino group NH, right pointing arrow basic. Carboxyl group Cu, right pointing arrow acidic. In acidic solution, low pH. Environment donates H. COO gains H, right pointing arrow becomes COOH, neutral. NH already protonated to NH. Small diamond form, positively charged ion. In basic solution, high pH. Environment accepts H. COOH loses H, right pointing arrow becomes COO. NH loses H, right pointing arrow becomes NH. Small diamond form, negatively charged ion. At isoelectric point pi. Amino acid exists as Zwitterion. COO and NH both present. Net charge equals 0. Zwitterion structure example. HN, CHR, COO. Internal transfer of H from COOH to NH. Summary table. pH condition carboxyl group amino group net charge. Acidic low pH COOH, neutral NH positive positive. Basic high pH COO. Negative NH neutral negative. Neutral pi COO negative NH positive 0 Zwitterion. Q4. What are essential nutrients? Why is it that certain nutrients are essential for some animals and non-essential for others? Definition of essential nutrients. Nutrients required by organisms for survival but cannot be synthesized by their own bodies. Must be obtained from the diet. Categories of essential nutrients. Essential amino acids e.g., lysine, methionine. Essential fatty acids e.g., linoleic acid. Vitamins e.g., vitamin C, B complex. Minerals e.g., calcium, iron, zinc. Water. Non-essential nutrients. Can be synthesized internally from other molecules. Not required in the diet e.g., glucose, some amino acids. Why essentiality varies across species. 1. Metabolic capabilities differ. Humans cannot synthesize vitamin C, so it's essential. Most animals, like dogs, can synthesize vitamin C, so it's non-essential for them. 2. Genetic differences. Genes encoding enzymes for nutrient synthesis may be present and functional in one species, absent or non-functional in another. 3. Evolutionary adaptation. Species evolved in environments rich in certain nutrients. So, biosynthesis became unnecessary, right pointing arrow genes lost over time. Examples. Nutrient essential for non-essential for vitamin C humans, guinea pigs dogs, cats. Taurine cats humans. Arginine cats humans conditionally. Summary. Essential nutrients equals must be eaten. What's essential varies by species. Age, health. Depends on enzyme presence, diet, evolutionary history. Q1. How does short-term regulation of urea cycle differ from long-term regulation? Urea cycle overview. The urea cycle, or ornithine cycle, is a metabolic pathway that converts toxic ammonia into urea. It mainly occurs in the liver. Urea is then excreted via kidneys. Short-term regulation. Short-term regulation means immediate or rapid changes in enzyme activity based on substrate or effector concentration. Key features. Acts within minutes to hours. No change in gene expression. Occurs by allosteric regulation. Substrate availability. Enzyme phosphorylation. Key regulator. N-acetylglutamate (NAG). An allosteric activator of carbamoyl phosphate synthetase I, CPSI. CPSI catalyzes the first step of the urea cycle. NAG synthesis increases when amino acids like arginine are present. Mechanism. Increased protein catabolism right pointing arrow up pointing arrow NH and arginine right pointing arrow up pointing arrow NAG right pointing arrow. Activates CPSI right pointing arrow accelerates urea production. Summary points. Quick response to nitrogen load. Involves enzyme activation, deactivation. Does not involve new protein synthesis. Long-term regulation. Long-term regulation involves transcriptional changes, influencing enzyme synthesis. Key features. Takes hours to days. Occurs via hormonal signals and gene expression. Results in increased or decreased enzyme levels. Mechanism. Prolonged high-protein diet or starvation right pointing arrow up pointing arrow transcription of urea cycle enzymes. CPSI. Ornithine transcarbamylase, OTC. Argininosuccinate synthetase. Argininosuccinate lyase. Arginase. Hormones involved. Glucagon and cortisol enhance transcription during fasting or protein-rich intake. Summary points. Slow but sustained regulation. Involves mRNA synthesis and protein synthesis. Ensures enzyme levels are sufficient for long-term nitrogen disposal. Comparison table. Feature short-term regulation long-term regulation. Timescale minutes to hours hours to days. Mechanism allosteric activation e.g., NAG gene transcription and translation. Enzyme activity modified temporarily changed through new synthesis. Key regulator N-acetylglutamate glucagon, cortisol. Response to immediate nitrogen load sustained protein intake, starvation. Q2. What are the factors affecting the rate of enzyme action? Enzyme action basics. Enzymes are biological catalysts that speed up chemical reactions by lowering activation energy. Their activity can be influenced by various internal and external factors. Major factors. 1. Temperature. Optimal range. Enzymes work best within specific temperatures, usually 37 degrees Celsius for humans. Too low. Molecules move slowly right pointing arrow low collision rate. Too high. Enzyme denatures, loses shape. Upward trend graph. Bell-shaped curve. Max activity at optimum temperature. Some enzymes need activator molecules to function efficiently. Q3. How amino acid reacts as acid and base. Introduction. Amino acids are amphoteric. They act as both acids and bases. They contain amino group NH, right pointing arrow basic. Carboxyl group Cu, right pointing arrow acidic. In acidic solution, low pH. Environment donates H. COO gains H right pointing arrow becomes COOH neutral. NH already protonated to NH. Small diamond form, positively charged ion. In basic solution, high pH. Environment accepts H. COOH loses H right pointing arrow becomes COO. NH loses H right pointing arrow becomes Q4. What are essential nutrients? Why is it that certain nutrients are essential for some animals and non-essential for others? Definition of essential nutrients. Nutrients required by organisms for survival but cannot be synthesized by their own bodies. Must be obtained from the diet. Categories of essential nutrients. Essential amino acids, e.g., lysine, methionine. Essential fatty acids, e.g., linoleic acid. Vitamins, e.g., vitamin C, B complex. Minerals, e.g., calcium, iron, zinc. Water. Non-essential nutrients. Can be synthesized internally from other molecules. Not required in the diet, e.g., glucose, some amino acids. Why essentiality varies across species. 1. Metabolic capabilities differ. Humans cannot synthesize vitamin C, so it's essential. Most animals, like dogs, can synthesize vitamin C, so it's non-essential for them. 2. Genetic differences. Genes encoding enzymes for nutrient synthesis may be present and functional in one species, absent or non-functional in another. 3. Evolutionary adaptation. Species evolved in environments rich in certain nutrients. So, biosynthesis became unnecessary right pointing arrow genes lost over time. Examples. Nutrient essential for non-essential for vitamin C humans, guinea pigs dogs, cats. Taurine cats humans. Arginine cats humans conditionally. Summary. Essential nutrients equals must be eaten. What's essential varies by species, age, health. Depends on enzyme presence, diet, evolutionary history. Checkmark Q1. How does short-term regulation of urea cycle differ from long-term regulation? 1000-line detailed answer style explanation below. Writing hand introduction to urea cycle. The urea cycle, also known as the ornithine cycle, is a metabolic pathway that converts toxic ammonia into urea for excretion in the urine. It occurs primarily in the liver. Urea formation is crucial for detoxifying ammonia produced from amino acid metabolism. Anti-clockwise arrows types of regulation. Urea cycle is regulated at two levels. Short-term regulation. Long-term regulation. Keycap 1 Short-term regulation of urea cycle. Test tube definition. It refers to the rapid changes in urea cycle activity that occur in response to immediate metabolic needs within minutes to hours. Gear mechanism. Regulated primarily by allosteric activation of carbamoyl phosphate synthetase I, CPSI, the rate-limiting enzyme of the cycle. Key regulator. N-acetylglutamate, NAG. Microscope role of NAG. NAG is an essential allosteric activator of CPSI. It is synthesized from glutamate plus acetyl-CoA right-pointing arrow NAG. NAG levels increase when amino acid levels are high, e.g., after protein-rich meal. Brain example. After a high-protein meal. Up-pointing arrow amino acids right-pointing arrow up-pointing arrow occurs rapidly within minutes to few hours. Pushpin summary of short-term regulation. Checkmark Q2. What are the factors affecting the rate of enzyme-catalyzed reactions? Presented in a comprehensive, diagram-supported handwritten style. Writing hand introduction to enzymes. Enzymes are biological catalysts that speed up chemical reactions without being consumed. The rate at which enzymes work depends on several environmental and chemical factors. Microscope main factors affecting enzyme activity. Keycap 1 Temperature. Thermometer optimal temperature. Enzymes have a specific temperature at which they work. Most efficiently called optimum temperature. Example. Human enzymes approximately equals 37 degrees Celsius. Up triangle increase in temperature. Increases kinetic energy right pointing arrow faster collisions between enzyme and substrate right pointing arrow increased reaction rate. Down triangle beyond optimum. High temperatures denature. Checkmark conclusion. Enzyme function is highly sensitive to changes in the environment. Understanding these factors is crucial in medicine. Hormones synthesized by anterior pituitary gland. The anterior pituitary, or adenohypophysis, synthesizes and secretes several key hormones. Growth hormone, GH, stimulates growth of bones and tissues. Thyroid stimulating hormone, TSH, stimulates the thyroid gland to produce thyroxine. Adrenocorticotropic hormone, ACTH, stimulates adrenal cortex to release cortisol. Follicle-stimulating hormone, FSH, stimulates follicle development in ovaries and spermatogenesis in testes. Luteinizing hormone, LH, triggers ovulation and testosterone production. Prolactin, PRL, promotes milk production. Melanocyte-stimulating hormone, MSH, influences pigmentation. 2. Regulation of respiration in humans. Respiration is controlled by medulla oblongata and pons, respiratory centers that regulate breathing rate and rhythm. Chemoreceptors detect CO2, O2, and pH levels in blood. Located in carotid and aortic bodies. High CO2 increases respiration rate. Low O2 or blood pH also stimulates respiration. 3. Hormonal regulation of ovary. FSH stimulates follicular development. LH induces ovulation and corpus luteum formation. Estrogen, produced by follicles, responsible for endometrial proliferation. Progesterone, secreted by corpus luteum, prepares endometrium for implantation. 4. Free radicals and their sources. Free radicals, highly reactive atoms, molecules with unpaired electrons. Sources: mitochondrial respiration, ROS. Pollution and UV radiation. Smoking. Inflammation. Certain drugs and chemicals. 5. Fat-soluble vitamins. Vitamin A. Vision, immunity. Vitamin D. Calcium absorption, bone health. Vitamin E. Antioxidant. Vitamin K. Blood clotting. 6. Bohr effect. Describes how CO2 and pH affect hemoglobin's affinity for O2. High CO2, low pH. Decreases affinity. Right pointing arrow releases more O2 to tissues. Facilitates oxygen delivery during high metabolic activity. 7. Urea cycle. Occurs in liver. Detoxifies ammonia by converting it into urea. Main steps. Ammonia plus CO2 right pointing arrow carbamoyl phosphate. Ornithine right pointing arrow citrulline right pointing arrow arginosuccinate right pointing arrow arginine right pointing arrow urea. Excreted via urine. 8. Water-soluble vitamins. Vitamin B complex. Energy metabolism B1 to B12. Vitamin C. Collagen synthesis, antioxidant. Easily excreted in urine, not stored significantly. 9. Factors affecting rate of enzyme-catalyzed reactions. Temperature. Increases rate until optimum, approximately 37 degrees Celsius. Higher temps denature enzyme. pH. Each enzyme has an optimum pH. Extremes denature the enzyme. Substrate concentration. Increases rate until saturation, Vmax. Enzyme concentration. More enzyme equals faster reaction if substrate is not limiting. Inhibitors. Competitive and non-competitive inhibitors slow down enzyme activity. Cofactors, coenzymes. Required for full enzymatic activity. Product concentration. End product inhibition regulates pathway. Ionic strength. Affects enzyme structure and function. Q. Describe the protein in terms of level of organization in primary, secondary, and tertiary structure. Introduction to protein structure. Proteins are complex biomolecules composed of amino acids linked together by peptide bonds. They play vital roles in virtually all biological processes, including structural support, enzymatic catalysis, transportation, signaling, and regulation. The functionality of proteins is deeply rooted in their structure, which can be organized into 4 levels: primary, secondary, tertiary, and quaternary structures. In this answer, we focus on the first 3. Primary structure of proteins. The primary structure of a protein refers to the linear sequence of amino acids in a polypeptide chain. This sequence is determined by the genetic code and is unique to each protein. 1. Amino acids, the building blocks. Amino acids have a basic structure: a central alpha carbon, an amino group NH, a carboxyl group COOH, a hydrogen atom, and a variable R group side chain. There are 20 standard amino acids, and the nature and sequence of their side chains determine the protein's properties. 2. Peptide bond formation. Amino acids are linked by peptide bonds, formed through condensation reactions, loss of water. Example: glycine plus alanine right pointing arrow glyalidipeptide plus HO. 3. Directionality. The sequence begins from the N-terminus, free NH group, to the C-terminus, free COOH group. Example sequence: Alagly S or THR Lu. 4. Importance of primary structure. Determines higher order structures. A single change mutation in the primary structure can cause serious diseases, e.g., sickle cell anemia. Valine replaces glutamic acid in hemoglobin. 2. Secondary structure of proteins. The secondary structure arises from hydrogen bonding between the backbone atoms of amino acids in the primary chain. It represents the localized folding of the polypeptide chain into regular structures. 1. Types of secondary structures. A. Alpha helix. First proposed by Linus Pauling. Right-handed coil resembling a spring. Hydrogen bonds form between the carbonyl oxygen of one amino acid and the amide hydrogen 4 residues away. Stabilizes the structure. Example proteins: keratin, myoglobin. Pleated sheet. Can be parallel or antiparallel. Chains lie side by side forming pleated sheets. Hydrogen bonding occurs between adjacent chains. Found in silk fibroin. C. Turns and loops. Connect helices and sheets. Found on the protein's surface and are flexible. Allow the polypeptide to fold into a compact structure. 2. Stabilization of secondary structure. Hydrogen bonding is the major force. Sometimes affected by proline residues, helix breakers. Influenced by pH, temperature, and sequence. 3. Tertiary structure of proteins. The tertiary structure is the three-dimensional shape formed by the entire polypeptide chain due to interactions between R groups, side chains, of amino acids. 1. Types of interactions. A. Hydrogen bonds. Between polar side chains e.g., O and NH groups. B. Ionic bonds, salt bridges. Between charged side chains e.g., lysine plus and glutamate. C. Hydrophobic interactions. Nonpolar residues e.g., valine, leucine tend to cluster away from water inside the protein core. D. Disulfide bonds. Covalent bonds between sulfur atoms of two cysteine residues. Provides extra stability e.g., insulin. E. Van der Waals forces. Weak attractions between close atoms. 2. Folding and shape. Folding is spontaneous but can be assisted by chaperone proteins. Globular proteins. Compact, rounded e.g., enzymes, hormones. Fibrous proteins. Elongated e.g., collagen, keratin. 3. Domains. Functional regions of proteins. Each domain can have specific activity e.g., DNA binding, catalytic. 4. Misfolding can lead to diseases like Alzheimer's, prion diseases, Parkinson's. Comparison of the three structures. Feature primary structure, secondary structure, tertiary structure. Description sequence of amino acids, regular folding, alpha helix, beta. Sheet 3D folding of entire chain. Bond type peptide bonds, hydrogen bonds, H bonds, disulfide bonds, ionic bonds, hydrophobic interactions. Stability, strong covalent bonds, moderately stable, highly stable, depends on environment. Function determines higher structures, provides local stability, defines proteins' activity and specificity. Examples. Lysozyme. Primary, 129 amino acids. Secondary, alpha helices and beta sheets. Tertiary, globular protein folding allows it to cleave bacterial walls. Hemoglobin, note, also has quaternary structure. Each subunit has tertiary structure that binds oxygen. Clinical significance. Mutations in primary structure. Cause misfolding, right pointing arrow disease. Example, sickle cell anemia, cystic fibrosis. Protein denaturation. High temperature, pH change, or chemicals can disrupt secondary and tertiary structures but not primary. Leads to loss of function. Experimental techniques to study structure. X-ray crystallography reveals 3D arrangement. NMR spectroscopy, solution structure of small proteins. Cryo-electron microscopy, visualization of large complexes. Conclusion. Proteins are dynamic molecules whose function depends on their unique structure, built step by step from the sequence of amino acids, primary structure, localized folding patterns, secondary, and the final 3D conformation, tertiary structure. Understanding these structures not only reveals the inner workings of biological systems but also aids in disease diagnosis, treatment, and biotechnology.